Hello, everyone. I'm Christy Risk, Senior Editor at Genome Web, and I'll be your moderator for today. The title of today's webinar is From Panels to Exome, Assessing the Technical Performance of dsDNA Capture Probes for NGS Variant Detection, and our sponsor is Twist Bioscience. Our panelists today are Dr. Robert Daber, President and Chief Technology Officer of Genosity, and Mark Consugar, Next Generation Sequencing Field Application Scientist for Twist Bioscience. You may type in a question at any time during the webinar. You can do this through the control panel, which usually appears on the right side of your screen. Click on the Q&A box in the control panel to ask a question. Please make sure the drop-down menu says All Panelists. If you do not see the Q&A box in the control panel, move your cursor over the main panel until you see the control icons on the bottom. Select the Options menu and then select Q&A. We will ask the panelists questions after the presentations have concluded. Dr. Daber, please go ahead.
Thank you. Good morning to those of you on the West Coast. Good afternoon to those of you on the East. Thank you to the Genome Web folks for putting together this webinar series. I've had the opportunity to participate in a few of these, and it's just a great forum to be able to bring advances in the field, recurring questions just to the broader community. I'd also like to thank the sponsors at Twist for giving Genosity the opportunity to talk about this topic and our experience with their Exome product. And so, as the title says, From Panels to Exomes, the main question we want to address today is whether or not the technology has hit a point where we can begin to think about performing exome sequencing and doing computational slices over designing and updating and constantly feeding and watering the concept of having smaller, more focused panels. And so this isn't a novel idea that Genosity brings to the table. This is actually something that has been kicking around for some time and being performed by many laboratories today. I want to give credit where credit's due. Sherry Bell, who I had the opportunity to work with in my last organization, was a huge proponent of this concept of really moving towards the use of an exome and being able to slice out other content. And so, as was mentioned, we'll have two speakers today. I'll speak on behalf of Genosity and our experience with the Twist product, and then hand it over to Mark, who's going to speak about some of the technical details about the Twist product line. And so the topics that I'm going to cover include the development and evolution of panels, specifically the process of developing a new LDT. Most of my career has been spent working in the clinical arena as next-generation sequencing has emerged as a technology platform there, and going through product life cycles of developing new products, new panels, and bringing them to commercialization in the clinical testing arena. And so a lot of sort of my focus will be on that approach and that process. That's not to negate the research applications where these technologies have tremendous value and tremendous power. The standards are just a little bit higher at the clinical side, and so as we look at technical performance, that is always the perspective that I'm taking when we look at next-generation sequencing or other technologies. After reviewing the process, we'll take a look at the requirements of what it would take to build something like an ultimate panel, the exome being that ultimate panel before we make a jump to a genome, and then spend the back end of the talk really describing our experiences with the Twist product, the Twist chemistries, as they relate to capture. And so Twist offers both custom panel design as well as exome or customized content on the exome. Our experience has been with their exome product, and so a lot of the topic will be just focused on that. So it's really important when we are looking at technologies to start with the entire process. And so here is the process really from the concept of I want to bring a test to market, or I want to develop a test, whether it be for research or clinical applications, all the way through that launch. It involves things like, before we're even talking about chemistries or sequencing instruments, what content do we care about? What are the genes and mutations we need to consider? What types of specimen sources do we need this chemistry and these technologies to work with? How do we assess the available options? And what's exciting is, even today, when next-generation sequencing is in its maturation process, and I think a lot of chemistries are maturing, we still see novel approaches and novel twists on old applications. And so there's always new technologies to sit down and think about. when we're talking about bringing on a new test or developing a new assay. After that, we begin to pivot into the laboratory process, where now is the actual evaluation process, optimization, and oftentimes there's a lot of refresh that happens during these stages. Sometimes two competing technologies or platforms will be used simultaneously in the lab while we're trying to optimize to see which one in the end will have the best performance characteristics to our acceptance criteria. After that, we make our final selection, we move on to an analytical validation and begin the testing process and the launch. And so I show these steps because it's not as simple as just pulling something off the shelf. Most of the time, it involves a lot of questions and digging and trial and error. And historically, this process is about 6 to 18 months. I think the fastest any team I've worked on has brought a new test to market is maybe 2.5 to 3 months. So there's a significant investment just for a single test, and this will become very important as the story evolves and continues. And so one of the first things we need to do is define what our intended use is. So this is really sitting down and looking at what is the content, the targets, the specimen sources, what types of mutations. Do we have fusions, and should we be thinking about an RNA-based approach in addition to a DNA-based approach, or can it be all DNA-focused? What are the limits of detection? Do we have heterogeneous samples, such as in tumors, really pushing the extremes in cell-free today with really low levels of sensitivity? Of course, cost is always important, turnaround time for the entire process, as well as expected volumes. And these are all key considerations as you sit down and begin to move into the design process. I always like to show this slide because even today, when we are 6, 7 years out from the first launch of a clinical next-generation sequencing test, when we sit down and say we want to launch a test in tumor profiling, and we begin to go through the exercise of looking at the literature, looking at some of the key stakeholders in the field, it still surprises me that the intersection, the content that everyone agrees as being necessary, is quite small relative to the total size. So when we talk about 400 genes across all the entities with all their expertise, the subset is still in the low 100 range. And the same thing on carrier screening. Today there's no commercial off-the-shelf products for labs to go and start carrier screening. All of the projects that I've been involved with has been a custom design, sitting down, looking at all the panels, looking at the literature, and trying to develop the content. And even here we see that the intersection is quite small. And so this becomes a challenge. We're going to sit down today at the beginning of this 6 to 18-month process, and we've got to select the content. And so what's challenging, and the risk we often take, is when we select that final content, there are genes in these other private circles. And so there's a risk between, do we include all 300 or 400 genes, or do we take a gamble and say that by the time we develop and launch a test, that one of these other markers is not going to emerge as now being absolutely critical. And so it's a lot of pressure trying to develop this content and stay ahead of it, which is where this concept of potentially moving to the exome starts to become quite attractive. After we've identified content, we now have to look at the actual technologies that we want to use, and there we start with, is this going to be an extension of something we've already done? Right? Are we going to be expanding our current panel with the next version? Are there any off-the-shelf kits available? And do we have to think about a de novo assay development? And based on the organization, whether it's a really small lab with minimal R&D expertise, or a big commercial laboratory where there's entire research and development departments, is going to depend on which of these avenues we're going to take. But either way, after we've decided on content, we need to think about the methodology. And here's just an example of some of the common methodologies that are out there. It's great. There's many options. There's a lot of competition, so it's constantly getting better, and technologies that were available years ago have really matured now, where there's a lot of equivalent performance, and we'll show some of that data in later slides. But either way, as a new lab coming to this question, and that's what Janasi spends a lot of time with now, is helping new labs get into the next generation sequencing game. This is a challenging question. What methodology should we select, and how do we assess it? So when we move to evaluating, there's a few key considerations. One is the availability of the platform or the technology, and whether or not there's any additional capital requirements. For example, 10x, if we wanted to begin doing longer read type library preps, there's some capital investment that needs to be considered, as opposed to just buying the chemistries off the shelf. coming into this is the cost of these library preps, ease of workflow. So ironically today, even though we're many years into next generation sequencing being common practice in laboratories, workflows have still not matured to the point of being completely turnkey, so there's opportunities there. They continue to get easier, but this is a key consideration. And then lastly, the performance itself. How well does the assay perform? And so key questions around the performance when we're evaluating a technology are around the specimen. So the specimens that I'm working with, I've got, I can't really control the input nucleic acid amount too much. Right? If I'm working with small FFPE samples or dry blood spots or oral rinse specimens, a bit more challenging. So does the specimen type for my indication of use actually fit with the technology? On the capture side, the first question is, can we even design a panel to capture the regions we care about? Right? This is the in silico modeling, or do we have to make concessions there? And then once we've got the chemistry, do all of those regions actually capture? And if they do capture, are they captured to sufficient depth to be able to confidently call positive and negative events? And then are we efficient with this process? So we may have a technology that captures all of our regions, gives us enough depth, but if we've got significant off-target or other challenging aspects, the cost of sequencing might be too high. And then finally, from an accuracy perspective, there's enzymes in the design of libraries. Enzymes have error profiles, slipping events, around indels, homopolymers. And so do we have confidence in the ability to make appropriate calls, or do we get left with false negative or false positive mutations? So to measure performance, we look at some key metrics. And the reason I'm spending time talking about this is really just to expand upon all of the efforts that happen when we are building a panel, right? Each panel has to go through these processes. And so the metrics that we look to optimize are things like the % usable data. When we sequence a library, we generate 10 million reads or 100 million reads. How many of those, after we remove off-target, after we remove duplicates, are actually usable? And how efficient are we in that process? How about insert sizes? In applications where the insert size is really important, how reproducible is it, whether we're looking at enzymatic or mechanical-based shearing? How many dropouts do we have across our target regions? How uniform is that coverage? And then again, these background profiles. And on the wet lab side in the chemistry, things that we look to optimize are DNA fragmentation and repair ligation, pre-capture, PCR capture, and post. And what I'm going to do is talk through some of these nuances because it's not a one-to-one relationship. And the reason I'm talking about this is our engagement with Twist started at the tail end of last year after we had spent a significant portion of time, led by Mike Moreau, our head of R&D, who was looking at, as Genosity goes out and tries to help laboratories make technology selections, as we build out some clinical research assays for clients to run in our laboratory, what is the best approach? And so we spent significant amounts of time looking at different enzymes, looking at different kits, looking at different capture conditions to really try and optimize this. And so that had happened for a good 7 to 8 months, and then Twist had asked us if we wanted to participate in testing out their Exome product. And so we did that on the tail end of all this activity and efforts. And so as I'll show you the data later, we were incredibly excited about the performance of their kit, but we had to go through all these exercises to really understand those nuances before we got there. And so here's just one example where optimizing something like DNA fragmentation can have an impact on multiple different metrics in terms of insert size, the number of dropouts indirectly, and then the error profiles. And so here's one of the key things when we look at informatics and the processing of data. So part of the informatics process, the pipeline, is aligning reads to the reference genome, and then we go through this realignment around local indels. And as you can see in the schematics on the left here, coming out of the aligner, we see many what would appear to be variants present in the data, but after doing a realignment, we see that there's actually an indel there. And so where this comes into play is when we talk about a mechanical shearing versus an enzymatic shearing. And what we saw is our pipeline got choked up, and instead of taking about 15 minutes for the step where we use a tool called Abra, it took about 960 minutes. And after going back and digging in, what we saw is that when we switched from a Covaris to an enzymatic shear, we saw a significant increase, a 10-fold increase in the number of interchromosomal mate pairs. And so this became a key consideration for us when we're looking at technologies as to whether we needed to look in somatic applications where we're looking for low-level, low-frequency events, is to use a mechanical shear as opposed to an enzymatic shear. So that was something we learned early on, and in our data later, we'll talk about how we're able to use both the enzymatic shear inside the twist workflow for germline applications, and then for any somatic applications, we can swap out and do a mechanical shear and continue on with their chemistries and still see the robust performance regardless of how we're shearing. But for us, this was key to move and make that switch. Another step to optimize is looking at adapter ligation, and this really comes down to an impact on how many cycles of PCR we're doing, what input amounts can we use, how does it affect coverage as well as duplicate rates. And here's just empirical data showing that both with 50 nanograms as well as 500 nanograms of input, we're able to get efficient libraries to be generated. And this is really critical. And this means that we can work with small amplica or small FFPE samples, small biopsies, or things like dry blood spots where the DNA yield is not very high, or routine blood or oral rinse type specimens. Lastly, here we'll just look at the impact of actual PCR itself, so the polymerases that are used and the number of cycles, the capture conditions, and then the post-capture PCR. And here the impacts are primarily going to be on the percent of usable data, so our on-target rate, our percent duplicates, and that will have direct ties on coverage uniformity and dropouts. And so here's real-world data looking at two polymerases. All things being equal, we see very different coverage profiles. So this plot is the count, the number of regions that have a given depth of coverage. And on the x-axis, we've got the region depth divided by 10. And so what you can see is that with an enzyme like kappa hi-fi as opposed to Hercules 2, we get a very different distribution of coverages, so different uniformity. In the ideal world, every region would be covered equally well. So the tighter this distribution is, the better it is in terms of overall assay efficiency and performance. Here's some data where, based on the HIBE, right, the number of samples you're plexing can also have impacts on uniformity. And then here's data just where we're actually switching out who manufactured the baits. And so I come back to this schematic because the data and all this optimization I just showed is really just part of the wet lab process. There's all of these steps. It takes 6 to 18 months per panel. And when we couple in all of the other activities that happen to commercially launch a test, things like building out IT workflows, working on all of the marketing and reimbursement test requisitions, and then the regulatory aspects around training and competency, there's a significant amount of work that happens outside of just the laboratory itself for every single test. When we couple this with the fact that the content is continually evolving, right? The number of genes in 2016 that now are more important in 2018, and this is just an example on somatic looking at myeloid. It's the same story in neurology, in cardiology, and others, where the important markers continue to expand, and so this system of constantly building and updating panels becomes quite challenging as well as expensive. When you look across the menu at a given academic medical center of all the different testing types, you now have this update and refresh across all of these different disease areas. And so it begins to really beg this question, right? The question that we want to address in this topic today is, are we there yet? Have we reached a point from a technology and a cost perspective where what we can perform is computational target enrichment? So if we look at the cost of exome sequencing and we look at evolutions of the sequencing platforms as well as the chemistries, we're now conceivably in a price range where you may be able to do whole exome sequencing at scale for under $150, under $200. And so it begs the question, is this now the approach? If the costs are there technologies, capture technologies that allow us to get the same degree of analytical sensitivity? And so what would be the requirements for something like an ultimate panel, an exome per se, that could be used for all of these different applications? And so these requirements are listed here on the right. There are others, but these are the primary ones. So first and foremost, do we get consistent coverage of our critical regions? So this is that reproducibility of capture, but then also the critical regions that we defined. Can we even capture them with the methodology? Is the methodology compatible with DNA from an array of different sample types? Easy ones in green, blood, bone marrow. More difficult ones in blue, oral rinse, dry blood spots, FFPE. Is it compatible with the range of input amounts, so from 50 to 500? Is it cost effective? And here we need to consider two prices. One is when we're capturing and we're trying to get the lowest performing or the worst performing regions above a minimum coverage threshold, are we doing that at the expense of sequencing costs? It's interesting. I sign out clinical cases from many different panel types over different technologies, and the number of germline panels that are being sequenced to 500 to 1,000x coverage just to get the worst performers above 20x. It's at least 3 or 4 different tests that I have experience with where that's the case. And so we pay for it in sequencing to get the coverage in those poorer regions. And then the library prep course, of course, and as well as error profiles of the enzymes themselves. So what I'd like to do for the next 10 or so minutes is now talk through our experience with the Twist exome. And as I said earlier, Twist has a variety of different options, capture panels from small panels to exome, and Mark will touch on those. But for us, the data that I'm showing is primarily around the core exome kit. So as I mentioned, last year we set out to really try and ask the question, what is the best type of approach from a library preparation and capture perspective? And so we tested all the main chemistries and kits out there, looked at permutations to try and really assess performance. If we drill into the capture side alone, when we talk about Twist and we look at a competitor such as IDT, the content is fairly consistent. In the Twist exome, we've got about 33 megabases of the genome covered, whereas IDT has 39. There's 0.4 megabases unique to Twist and 6.2 unique to IDT, with an overlap of about 32.6. And so all of these analyses going forward are going to look at that overlapping region so that this way, if there are regions unique to the IDT exome that are more problematic, it doesn't get penalized as we start to look head-to-head at some of the comparison in our hands. So here's a first slide of some real-world data highlighting our experience. What we've got is both enzymatic and covirus shearing techniques. We've got different library prep chemistries as well as the capture chemistries, and we've looked at a few different plexing ratios with the different baits. And so what we didn't do in this figure here is normalize for the total input of reads. So we didn't set the denominator at 41 or 33 million reads and normalize coverage and performance to that. Each of these scenarios were run with the same input DNA amount. So we started with 500 nanograms for everything, except for the Twist chemistry, which had an input of 50 nanograms. So I just want to highlight that nuance that everything else here is 500. Twist was at 50. And when we ran the Twist protocol, we didn't deviate. This was running straight out of what is recommended, whereas with each of these other conditions, we've taken some liberties to optimize based on a variety of different experiments performed before that. So if we look at these metrics at a really high level, what I've highlighted in red here is just some nuances or differences. And so what we have is slightly lower on target from Twist compared to our experience with IDT, so 90% versus 76. Dupe rates all well below 5, with Twist coming in at 2.8%. And this is, again, remember, with 50 nanograms of input DNA, we're typically the lesser amounts of input DNA, the higher the dupe rate. So we were very impressed with that. Overall, the other metrics are fairly consistent in terms of percent usable, somewhere between that 70 and 85% range, and mean coverages. Where the data really began to impress us is when we look at the actual percent of the target region that is covered to a minimum threshold. So what I've got highlighted in green here is anywhere where we had over 98% of the target region covered to that minimum coverage threshold. So when we look at 20x coverage or 25x coverage, these were the conditions that provided us with that type of overall coverage. And so for anyone who's been doing exome sequencing and has watched the evolution of this, we were once upon a time happy with 85, 90% of the target region covered to that 20x mark. Pushing to 95% is, really kind of earth-shattering. And now to begin thinking about an off-the-shelf kit that could actually hit 99% is pretty amazing. And so this is just looking now deeper in our experience with Twist itself. So here's a set of germline samples. What you can see is the consistency from the data. So in our hands, taking a set of real-world samples derived from either blood or oral cavity specimens, what you can see is the on-target rate is very consistent, as is the percent duplicates, as well as mean coverage, and the insert size. So for an enzymatic shearing-based approach, to see the insert sizes be this consistent within a few base pairs of each other is pretty amazing. And when we look at the uniformity within a high pool, we saw very consistent coverage. And so for anyone who's done a lot of hybridization-based library preps, Hive capture, you know that there's the quantification step that happens going into Hive, and there's some variation there. And even with the best of hands, oftentimes you can see some differences in uniformity from sample to sample. But here we saw almost spot on perfect, which is an attribute of our scientists who did the work, but also for the chemistry itself. And I'd also like to highlight that this was with about 187 nanograms of library into the 8-plex. This is contrasted to our other workflows where we're at about 500 nanograms. And so that difference in and of itself allows us to begin thinking about lowering the number of PCR cycles, or when we're working with low input samples, of being able to still go into Hive. And so that's really important because PCR, while it's our friend in molecular biology, it's also an enemy. We don't want to overuse it because there are implications related to coverage uniformity as well as artifact risks. Given the low dupe rate, we went ahead and looked at a sample that we had sequenced really deeply, and then downsampled that. And what we wanted to do here was ask the question, are we saturating in terms of our library diversity? And so from as low as 20 million reads all the way up to 120 million reads, what we saw is a linear relationship between the duplicates and the total number of reads. We don't see this kind of exponential growth in percentage of duplicates indicating that we've hit a ceiling, which would happen if we didn't have very good ligation efficiencies, right, where the library diversity was limited. So this was great. It showed us that we had a really good linear range as it relates to our percent duplicates. We also went ahead and performed some library preparations on FFPE samples. And so here's just highlighting one of the key metrics. Anyone who's done library prep on FFPE knows that it's a complicated material. When you perform library preps, typically things like percent duplicates begin to jump through the roof. And so with 100 nanograms, we upped the input FFPE versus 50 nanograms of genomic DNA. We saw that our dupe rates really hung in that 2.5 to 3.5% range, which was fantastic. If we look at target coverage now across all those different germline samples that we had shown the data for previously, we see that every single one of those samples hit a minimum of 99% of the target region being covered to 20x. And this is with about 60 million reads or 8 gigs of data. And for the 25x coverage, again, all but one sample hit that 99% mark. And so to me, these are just fantastic metrics for a library prep chemistry and capture chemistry. If we look at the data for the FFPE samples relative to the germline, and we look at, again, our percent covered on the y-axis and total gigs of data, we see in the blue the germline samples all cluster really well. They all had that 99% mark, whereas the FFPE samples are a bit more scattered. So digging into this a little bit more and beginning to do some downsampling experiments, we wanted to understand whether or not we could explain what was happening on the FFPE side. And so here's an experiment taking that really deep sample and downsampling from 120 million reads down to 20 million reads, and looking at the percent of the exome that's covered. And so what you can see is at both 20x and 25x, as low as 60 million reads, somewhere around that 60 million read part, we're still above 99% covered. And then we can begin to see the coverage drop pretty significantly around 20 million reads, which is quite small for an exome. And then what we did is we took those curves and we went back to the FFPE and germline data and plotted it. And what we see is there are several FFPE samples in the green circles that fall right in line with those downsampling models, meaning that the lower percent to 20x was actually due to just not having enough data generated, while the other ones circled in red, the other FFPE circled in red, they all fell in a single Hive reaction and indicated to us that perhaps we had some kind of technical challenge with that hybridization step for those set of samples. And so this is really promising. We've got some ongoing experiments now looking at real tumor specimens across this panel, again, pushing now the coverage up even higher as we generate more data required for somatic type applications. Here's real-world data for us in this downsampling arena, looking at the performance of the twist chemistry with the twist library prep versus our optimized chemistry and optimized version of IDT. And so on the right side here in the figure is the percentage of the target regions covered to 20x. And what we can see is when we get to about 15 gigs of total data, both applications are in that 99% range. However, with our IDT experience, we start to see 98, 98, 95% as we get down to that 6 gig part. And so what I'd like to do is just, for research applications and for other comparative technologies, I mean, 95% coverage at 20x is still very good. And so this isn't to discredit an alternative methodology. We're still very happy with that. But it does really highlight one of the benefits. And in the clinical space, that last 1 or 2% begins to have a couple different implications. And so first is how much data do we need to generate to get to that 99% mark? If we could generate that data at, say, 8 or 9 gigs as opposed to 15 gigs, well, that could have some cost implications. And then second is as we begin to look at some of the actual slices or content, how does this play over into covering the critical regions? And so another metric that's really important to look at is not just what I have shown here, which is covering to the minimum threshold, but as we try to push to covering regions to that minimum threshold of 20x, what happens at the other end of the spectrum? Do we have any targets that begin to over-capture, over-sequence? And so that's what this data shows is that, at some of these higher gig ratios where we need to get up to maybe 12 or 15 gigs to get 99% covered to 20x, we now start to sequence 1, 2% of our exome to well over 500x. And this has cost implications. And if we look at this slide here, tried to model out what it looks like to sequence exomes on a NextSeq or on the NovaSeq platform with one of the different flow cells, S1, S2, or S4. And in the blue lines, blue vertical lines in the graph on the right, we can see that at about 8 gigs versus 16 gigs, we begin to have some significant differences in overall sequencing costs. The table on the left summarizes this. So if we take the best case scenario and say, 8 gigs of data on an S4, we're really talking about a sequencing cost down to as low as $82 and fitting about 375 samples per flow cell. If we wanted to set a mark at 99% covered to that 20x threshold, alternatively, the cost doubles and the number of samples decreases by a half if we need twice as much data. And so this is just a key consideration, and this has to do with the uniformity of coverage, right? So again, the tighter the uniformity between the poorest performing region and the highest best performing region, the better we can actually contain costs. So the last thing I want to just highlight is looking at the genes of interest. So if we were to take a very conservative approach and cast a wide net, saying if we look across a space and take the average of what people are doing in terms of research, clinical research or clinical panels in cardio, inherited cancer, metabolic disorders, neurology, somatic tumors, or in carrier screening, we see that we range anywhere from low 70 to many hundreds to thousands of genes. The megabase sizes from as small as 0.28 megabases to as big as 7.1. And so what we wanted to do with these genes of interest slices is look at the performance of the twist exome. And so this is real-world data from downsampling a sample to 60 million reads, or about 8.9 gigs of data. And what we can see is that for all of these different indication slices, with the exception of tumor, we are hitting a mark of 99.6 or greater percent for 20x coverage and all over 99% for 25x coverage. So this is incredibly exciting. What this shows is that we can really begin to think about covering very big panels of genes with a single test. Tumor is interesting, and we need to go back and dig in, and perhaps there's some genes that are just a bit more problematic. The tumor stats here are falling a little bit below even what the whole exome saw. But for the other germline-based indications or genes of interest sets, we see very, very good coverage. If we drill in on something like an inherited cancer slice and look at the regions that failed, we can gain some even better insights. So out of this list of, say, 20 or so regions that had one position in the region fall below the minimum coverage threshold, what we can see in the table in the column all the way to the right is that we still have coverage of these regions. It's not as if they are dropping out entirely. So for a sample obtaining about 100x mean coverage, we're seeing somewhere between 7x and 19x at least one spot in these regions. 5 of these regions have homozygous deletions, so they're being flagged inappropriately. The region actually has a mean coverage of, say, 100x or so, and half of them, what's missing the coverage threshold is just the last few bases right before the end of the region itself. And so to go back and pad these regions is not a significant effort. It's not like we've got big holes in genes that we need to worry about and address. So if we come back now to this initial checklist, we can check almost all the boxes. What I didn't show here in the talk and didn't feel it was appropriate to check the box is just looking at the error profiles. I can tell you from our empirical data, we don't see any problems with it. I just didn't have time here to discuss and summarize all those results. So before I hand this over, just several conclusions. One, for anyone who's been involved in assay development, it's a known, very laborious process. It takes a lot of time. And oftentimes new markers are emerging at the tail end of a development project and makes it challenging to stay ahead of. Using the exome as an ultimate panel is really closer to a reality due to things like these robust chemistries as well as decreasing sequencing costs. And then specifically as we relate to the twist capture baits and the twist assay, we saw superior uniformity of coverage, and this was right out of the box. We didn't do any tweaks or modifications to the assay, whereas we had spent many, many months trying to optimize the other approaches. And then we had over 99% coverage of all these critical regions with 50 nanograms of input DNA. And so when we saw this, when we experienced this, we got really excited about the potential of what could be done here and whether or not the reality of moving towards a single exome and being able to do computational enrichment is closer than we thought. And so with that, I'd like to thank everyone who's been listening here today, and special thanks to the Genosity team, Mike Moreau, who did a lot of the R&D work, really trying to dig into the weeds of the exome. And so with that, I will hand it back over to Christy.
Thank you, Dr. Daber. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We will conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Mr. Kronsugar. Please go ahead.
Hello, thank you very much for everyone who's in attendance, and thank you, Bob, for this wonderful presentation that you had just given. And my name is Mark Kronsugar. I am a next-generation sequencing field application scientist with Twist, and today I'd like to talk about how Twist is improving the performance, efficiency, and flexibility of targeted sequencing. So to start, I just would like to give a summary of the 2 most conventional modes of next-generation sequencing for DNA. Here shown on the left is what is conventionally known as whole genome sequencing. It's very comprehensive but has a tendency to be very expensive. And then on the right, you're seeing what's known as targeted NGS. This also has a tendency of being very focused. It's less costly and can provide deeper coverage, because what you are doing is you are taking a portion of your genome and you're sequencing that selectively to deeper depths. In 2014, Minard et al. Had identified that there was some inefficiencies between whole genome sequencing and whole exome sequencing, which result in a sequencing sensitivity gap being generated. And what you can do is you can see at this line depicted here in this graph at 95% sensitivity that there's a white space which has been generated between the whole genomes depicted here in purple and green and the whole exomes which have been depicted here in orange. Now, one of the reasons for this gap that was generated is because a lot of the target enrichment pools that are available on the market have a tendency of having biased oligos in them. And to depict that, I'm showing this slide here. And what I'd like to show here at the very top is that all oligos start the same way. It is standard phosphoamidite chemistry, which is going to be used to synthesize your oligos, and they will be generated in a very uniform profile, which is not going to be dependent upon GC content whatsoever. However, the concentration of this seed oligo pool is not sufficient to really do much downstream applications, including next-generation sequencing, target enrichment. So the next step that has to be implemented is taking the seed pool and amplifying it. And this is the stage at which you can start introducing bias into your pool, because the amplification process is going to be dependent upon the GC content of a given oligo or oligo set. And the bias, as a result, can then translate into non-uniform sequencing off your sequencer. Conversely, on the right, I'm showing that Twist Bioscience has come up with a proprietary method to maintain the uniformity from the seed synthesis through amplification into the oligo pool, so that you have that uniformity translated from seed to pool, and then, as a result, a more uniform capture pool is going to translate into a more uniform sequencing. So the Twist advantage to using probe pools with minimal enrichment bias is that you're going to get a more uniform coverage distribution. And the more uniform coverage distribution I'm going to show in this graph here on the right. Now, there's two exemplars. One is Twist being in green, and the other is a competitor in gray. And the key is that you want to stay on the 100% base covered plateau for as long as possible, and then fall to zero as quickly as possible. By staying on the plateau longer here in the upper left, you're going to see improved coverage. The quicker that you fall off the plateau to zero, though, is going to result in a reduction of overrepresented reads. So when you compare the green coverage distribution line, which is Twist, to the gray of the competitor, all the shaded regions are translating to this improvement in uniformity, which is going to result in improvement in sequencing and a more efficient sequencing off your sequencer. So that all being said, Twist had gone back to see how now our improvements in probe synthesis and workflow had translated to trying to narrow the gap between whole genome and whole exome sequencing, which I had shown a couple slides earlier based on MineArts data in 2014. And what you can see now, when we use the NA12878 sample, the HapMap sample, is that Twist Bioscience has been able to narrow, if not remove, the gap that we had previously seen, such that the whole genome sequencing and whole exome sequencing from Twist, their lines run pretty much right on top of each other. To dive in a little bit deeper, I would like to show some data that was generated externally, which compares the Twist whole exome to other exomes on the market. Twist is shown in blue or dark purple, and the other exomes are shown in yellow, green, and red. And what you can see here on the right is that Twist by far has a very low duplicate rate, which had been echoed by Dr. Daber in his presentation. Conversely, we're showing with Twist Bioscience a superior sequencing depth across a varied amount of sequencer outputs relative to other exomes that are on the market. So whether it's the duplicate rate being superior or the coverage being superior across these sequencing outputs, Twist is definitely in the lead. The next slide I want to show, I understand it might be a little bit busy, but it's going to highlight how both configurations the Twist offers in the exome, be it singleplex or multiplex, is outperforming competitors that are also in the market. So on the far right here in green, I'm showing some targeted percentages at 20 and 30x for Twist's core exome at 1-plex and singleplex format. The first thing that should jump out is that there doesn't seem to be too much of a difference in performance between our singleplex and our 8-plex configuration, which is the case. And what we like to say is that customers will get singleplex quality data with Twist exomes in an 8-plex configuration. And this can have a lot of downstream positive effects in workflow. But before we get to that, I'd like to then compare these two configurations to one other competitor, his competitor I. Now, in both the singleplex configuration with Twist relative to the singleplex configuration with competitor I, or the 8-plex configuration between the two, Twist is outperforming both at 20 and 30x. And then finally, with competitor A in their singleplex format, you can see that Twist also outperforms in both its singleplex and its 8-plex form. So once again, just want to highlight that you can get singleplex quality data using an 8-plex pre-hybridization capture configuration, which means in a single hybridization reaction, you are going to get 8 exomes rather than a single exome, and that can have a lot of streamlined effects with your workflows in laboratories. Before I end my presentation here, I'd like to discuss real quickly that Twist also offers custom target enrichment designs and probes for custom projects. We will accept any design from or any region of interest from a customer if you provide it as the chromosomal regions as well as the build that you want as your reference. Within about 24 to 36 hours, we can turn around that design for the customer, have them review it, and once the design has been signed off, we will take that design intake and be able to synthesize oligos and deliver them to the customer in anywhere between 2 to 3 weeks. This very quick design turnaround time and quick synthesis turnaround time is going to enable our customers the ability to test, evaluate, and optimize their panels to get that final optimized design that they would like to scale up without necessarily having to commit an awful lot of financial or reaction resources. A lot of our customers are taking their design, doing a couple of pilot iterations of 16 hybridizations, tweaking it, tuning it with some assistance with Twist, and once they feel like they've zoned in on their design of interest, they shoot it off as a final design and then do the scale up. So it becomes a very time and cost-effective way of taking a custom panel, vetting it for its ability to be stable, and then having its performance assessed before you commit more dollars and more time to implementing it into your laboratory. I'd also like to show here that Twist offers their target enrichment products in a varying amount of configurations or kit configurations. From here on the left, the complete kit, which includes the library construction, adapters, blockers, buffers, beads, and probes, all the way down to probes alone on the far right. And we're willing to work with our customers to try and accommodate any kind of configuration that lies in between, so as to make sure that there is the least amount of disruption into their current workflow to allow them and enable them to get the best probes out there to do the target enrichment that they want to use to obtain the most efficient and uniform coverage off their sequencer. So in closing, I'd like to just highlight some of the Twist NGS target enrichment benefits, which includes our exceptional performance because of our double-stranded DNA oligo probes. Flexibility, we are agnostic with a number of library construction methods and target enrichment methodologies. And finally, this idea of maximizing your sequencer output so that you can reduce the number of duplicates and non-informative reads so that what comes off the sequencer to the best of its ability is going to translate to usable data to make the variant calls that are necessary for your research or for your customers. So with that in mind, we at Twist like to ask, why would you design tomorrow's experiments using yesterday's tools when we feel that the oligo probes that Twist can offer now are today's tools? And with that, I'd like to say thank you and return this to Christy for any questions.
Thank you, Mr. Consugar. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to give us feedback by taking our exit survey after the webinar. And now we'll start the Q&A. Bob, the first question is for you, actually a series of questions about how you treat FFPE samples. First, do you always perform DNA end repair for FFPE samples, and what kit do you use for that? And second, how many total cycles of PCR do you usually perform pre- and post-capture for FFPE samples?
All right. So for FFPE, we don't perform anything special during or after the isolation itself prior to library prep. So we'll isolate nucleic acid and then take it through Kovar shearing and then the standard end repair adapter ligation. So nothing additional done pre-next generation sequencing. And in terms of the number of cycles, it depends on which of those conditions we're talking about. And it's usually 10 cycles pre and anywhere between 8 and 10 cycles after.
Great. And Bob, are your capture probes double-stranded for both the whole exome sequencing panels and the targeted panels?
I believe that the question probably for Twist, and yes, it's the same. I could speak on their behalf, right? Same design for panels or exome.
Mark, do you want to jump in on that?
Yes, that is correct.
Okay, great. Bob, we have some questions about the comparison work that you did. First, when you compared IDT to the Twist captures, were they both at 8-plex per hybridization? And second, have you evaluated uniformity of coverage of different exome sequencing platforms?
So to the first question, we performed in the data represented here, it was a 4-plex with IDT. And an 8-plex with Twist. We ran Twist native to its protocol. We have done experiments going as high as 16-plex with IDT. We see some variations. We feel pretty comfortable we can get up to an 8-plex with that chemistry as well. But what was shown here was 4-plex. And then in terms of the uniformity, yes, we did. We looked at uniformity across each of these technologies that we investigated. We did the most work in comparing what we considered our final library prep conditions with IDT capture versus Twist, because we started doing this Twist work kind of at the tail end of our R&D, so we didn't go back and do extensive work and comparisons with some of the other methods that we displayed in the tables here.
Okay, great. Mark, can the Twist exome kit detect microdeletions and duplications?
So I've been working with a couple of investigators which are looking to answer that question directly. I personally have no experience seeing any of that data myself. I'm not sure if Bob has seen anything on his side or has assessed for it. Have you?
No, we haven't. And I don't know that the current design has sufficient coverage. There's several that we might be able to look at, but that would, my opinion is that would be an additional set of spike-ins if you really wanted to begin thinking about an exome replacing something like a SNP array or array CGH.
And Mark, the next question is also for you. Can the Twist workflow be automated with robotics?
So yes, that's a very good question. Twist is currently working with a number of the key automation vendors right now to establish this workflow. We are in as early as water testing with some, and in as late phase as doing actual library construction with others to sequence. So I would say by a slow rollout between now to the end of the year, we'll be getting ready to actually be doing some alpha and beta releases for testing with automation. That is our goal, is to actually have this whole system automated and offered in modules so that you could start it at any given point, given that a customer's workflow might not be our total workflow.
Okay, thank you. The next question could actually go out to both of you, so feel free to jump in. How do you make sure that the double-stranded DNA probes don't re-anneal to themselves?
So I can start with that one. So in the protocol, we use something that's called a two-pool melt. So what you do is you take the genomic DNA library and you melt it, and then you take the probes and you melt them separately. You snap cool the probes and then you mix them together, and the thought is that those probes are going to find their complement faster than their complement of the genome or the genome library than they're going to find necessarily their complement themselves at 70 degrees.
Bob, anything you want to add to that?
I don't know the full design of the probes and whether or not they're complementary across both strands, and so there's some thermodynamics clearly at the annealing temperatures that would favor binding to genomic DNA if they're not completely complementary, as opposed to having partially hybridizing to part of an opposing probe. So I think this is more technical on the twist design than I can comment.
Okay, great, thank you. And Bob, going back to you, on slide 39, you had a chart that mentioned percent on target, and we had a question about how you calculated the percent on target metric for the twist exome panel.
Yeah, so that had slight padding on it. We went out about 100 base pairs off of the actual target regions themselves, so some small padding.
Great, thank you. And what was the read length that you used for the exome sequencing? Our listener is assuming 150 base pairs, but is that correct?
That is correct, paired end 150 by 150.
Great. And which enzymatic fragmentation method did you use?
So Twist has it in their kit, and then in our evaluation, we had tested Kappa Hyper Plus, as well as Archer has an enzymatic shearing enzyme that we worked with.
Great. Mark, going back to you, will automation be available on the Agilent Bravo automated liquid handling platform?
Yes, that is something that we are working on scripting ourselves.
Okay. And the validation of next generation sequencing will still follow the guidelines. Will that still follow the guidelines of AMP and CAP that were published in the Journal of Molecular Diagnostics in May of 2017?
Yeah, so those guidelines are great guidelines, and then depending on the actual state that a laboratory is in, there may be additional guidelines from New York or from other regulatory bodies. So it really depends upon where the laboratory is accredited.
Okay, great. Bob, you had some data that showed that Twist had some lower on targets as compared to some other methods. And Mark, you might want to jump in on this. The question is, are there some modifications that can be made to the Twist exome that could improve those metrics?
Yeah, of course. So, what we showed was kind of an unbiased view of just running the assay without any tweaks or modifications. The other data we had was after months of us tweaking certain things around the volumes used and the conditions used during HIVE, and so I think that there are some tweaks that we can bring to the protocol. Overall, we still hit those beautiful percentage of target regions covered. So it wasn't too alarming to see the on-target be slightly lower, given that it still outperformed.
All right, great. Thank you so much. Oh, sorry, please go ahead. Yeah, and I think the one comment I'd like to make, I do know that the slightly lower on target or on target in general has a tendency of being a stat that a lot of us like to hang our hat on. But I think the one comment I would like to make is that I think at the end of the day, uniformity is where a lot of that loss in on-target, that slight loss in on-target is going to be well recouped on the uniformity side on the back end. When you're seeing uniformities that are going to allow for that kind of coverage, I think that's where it lies.
Absolutely, and also the percent dupe, right? So at the end of the day, you start with a total number of reads, and then you have to align them, remove off-target, remove duplicates. And so having a low dupe rate also compensates for any additional off-target, at least in the data that we have.
Great. I think that's a perfect note to end on, and that's all the time we have for today. We'd like to thank Bob Daber, Marc Sugar, and our sponsor, Twist Bioscience. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.